No. Awesome. Okay, welcome everybody. For anybody who did miss this morning, I'm Sam. I'm a medieval bioarchaeologist and a research associate at the School of Archaeology at the University of Oxford at the moment. And today I'll be talking about open methods. Some of what I'm going to be talking about really overlaps, obviously, with what a lot of other people are talking about today. But I'm going to be focusing more on open methods in terms of literally the sort of desktop-based or lab-based or fieldwork-based methods, the stuff that you actually do to kind of generate your data. But I'll touch a bit on analysis as well, but that's really Go-Young's realm later on. So where do open methods fit within sort of the umbrellas that Emma was talking about earlier? Really, it's over here, this nice sort of red polygon there. So it's about sort of developing standards to collect your data, however that might happen. And the metadata, and as we've sort of heard earlier today, metadata is just any of the data that describes your data. So how you got it, what it is, where to find it, how to use it, to allow for easier interpretation and for people to follow your decision-making skills, but also reproducibility. So somebody can literally kind of look at it like a recipe, follow what you did and understand how you got the results you got, and hopefully also do it again themselves. And this obviously also requires things like open source practices, open resources, and open data, because you need the data and the resources to be able to sort of follow the methods. So it all ties in together. It doesn't stand alone by itself. So why make your methods open? So apart from all of the fantastic reasons that Emma and Leggett have already talked about, there's some things specific to the methods that I think are quite important to kind of remember. I'll start with the sticks. So particularly for anybody in Europe, as Emma's already touched upon, big funders, particularly the European Research Council or the Wellcome Trust, who do sort of fund biomolecular and sort of health-related archaeology, require it. So if you want to be funded by these big funders, or you were already funded by them, you have to do this. And they might penalize you later for future grants if you haven't done it, or possibly have other sort of things in place to penalize you for this. So that's pretty much enough said on that. But there's other things in terms of publishing. Like we said, publishing is starting to change. Legging's already said that quite a lot of reviewers say that they want to see your code or your methods. So you might risk not getting published in certain top tier journals because some of them make it a requirement. So it's a really good thing to kind of have from the get-go, so that you can automatically go into those publishing avenues. And this being said, you're less likely to be reproduced by other people and therefore cited if they can't follow your working. So this is really kind of, going back to that sort of high school thing of maths, of put all your work in there so we can work out where you got to get your results. The carrots, the good things, transparency and reproducibility, and making them all open and accessible for other people to use. But also kind of what we touched upon in terms of people's ideas about scooping and things, it's staking a claim. If you have your methods open, people can see exactly how you did it. It's yours, they can cite you. It can allow for collaboration, like we've heard about already. So if you kind of have that out there, other people might say, oh, actually, if you add this extra strand to this method, or if you do this, let's work together, and it can improve your research. Reuse also always leads to citations, and I showed some really excellent graphs about how making things open just sort of boosts that. But also with some of the open methods resources I'm going to show you, it will often actually allow for review of your methods before you actually start the study, in line with sort of how healthcare research sort of operates, to allow you to improve your research methods before you even actually sort of start data collection. And often when you sort of have this pre-registered and pre-reviewed methods, it will lead to faster publication with different publishers later on down the line. And yet again, just more citations. It's just great. More people see it, more people reuse it. It's all good. So metadata is something that we're all going to probably be just badgering you with all of today. And if anybody kind of wants more information about exactly what metadata is, how to maybe structure it, how to start writing it, this is a really good article that I'd point you all to. And as you can see, this came out in the 1990s. So this isn't really a new concept, and ecologists have really kind of been hot on this for a while, but I think it's starting to trickle through into other strands of research. And essentially what this sort of talks about is when you're doing your own sort of research, and you've got your project here on the left-hand side, you should sort of start writing your metadata alongside it at each step. So you should kind of document your thinking and what you're doing. This can either be digitally or it can just literally be in a notebook. That's quite often how I sort of sketch things out first. So it then equates to something that you should be giving people in a metadata class. And this flows all the way through from you just designing what you want to do to the actual field and laboratory procedures you conduct, how that data is actually sort of physically collected, where you store it and enter it, and any quality assurance and quality control that you put that through. The actual sort of analysis of that data, which, as I mentioned, will sort of be in the next talk after lunch in more detail, but you should also be collecting metadata about your methods that you're using there, as well as metadata about your publications. So where you have published this, if you then decide to sort of separate your data out from that, and you should collate all of these different sort of bits and pieces. So what the data is, what your research workflow is, the overall project description, any sort of specific sub-projects you've had there, how accessible it is, and anything weird that you found during your research as well. And this all leads to secondary use and reuse. So you can see here that sort of each type of metadata can be used in different ways and flow back into the research framework. So I know a lot of people are quite keen and sort of, okay, well, I can document everything I do and kind of just keep a track of what I've been doing and then maybe report it somehow, but exactly how do I report that and publish that and get that recognition for, you know, trying to do the right thing and make everything open? I think we've all shared a lot of PLOS because they've got really good infographics for this. And there's a couple of different ways and a couple of different stages in your research where you can share and make your methods open. So I'm going to talk a bit about each of these in the sort of upcoming slides, but there's registered reports, which basically gives you, at least in the PLOS model, 2 separate publications where you do one before you start actually collecting your data, and then you're guaranteed a linked actual publication of results afterwards that's linked to sort of the research design. You can do a study protocol, which is more about exactly how the whole overarching study and research program is going to go, and exactly step-by-step how you're going to do everything, not just what's in the lab. If you're doing new method development, you can do a method article. That won't be applicable to all of us because some of us are just using sort of tried and tested methods. But if you are innovating in this way, that's obviously a really good way to make that sort of open, usually with your results to show how the methods actually work and can be validated. And then, of course, there's lab protocols, and this is a really great sort of emerging sector across all different types of science and sort of digital humanities as well, where you give people a step-by-step recipe guide. And I'll show you examples of what this all looks like. You can see here at what sort of stages in your research these things can kind of come out. So pre-registration and registered reports. So registered report is the actual sort of publication that comes with pre-registering your research. This has been most common in sort of health science and medical research, because usually you have to pre-register with the body to make sure the ethics are all okay and you're allowed to do this work usually on people. My partner works in clinical research, so this is something I hear about a lot. But this is something that's now coming to the fore in archaeological research, which is really, really great. And you can see that essentially it's to peer review before data collection. So I think any of us working particularly in the academic sector, we're quite used to this. If you want to apply for grants, you know, people are going to peer review what you want to do and decide if it's worthy of money. But this is a way to also get that published. So you can either do this before you've had that grant application, or you can do it after that grant's already been awarded. So the idea is you kind of develop your idea, you work out sort of what your research questions and things are, and then you get that editorial and peer-reviewed feedback there before you do your science. So it's where you deposit and publish those research questions in your study design. And this means that you get publication regardless of results. So this should hopefully avoid confirmation bias. So I think we're all kind of used to sometimes seeing really big, sexy sort of headline science being published, but you kind of go, are those methods actually appropriate for that? Or have they kind of changed that based on the results? But this avoids that sort of problem there in confirmation bias. So like I said, with PLOS, you get 2 publications. Some of them you only get 1, where you kind of pre-register it. There's kind of like a mock-up of your research design, and then you get 1 big publication at the end, or you get the sort of 2 where you get the registered report about, this is how I'm going to do it. This is my plan. I'll see you in a couple of years. And then you get the later publication after you've done it all to go, it worked or it didn't work, but regardless, here's the results. So generally this leads to much quicker publication, because you've already pre-registered and they've already peer-reviewed how you did it. It's then just about the results later, regardless of if they worked or not, which is really what science should be about. Obviously, this won't be applicable to all types of archaeological subdisciplines, because sometimes we just get what we get and you dig a site and that's just what there is. But for people who are maybe doing more lab-based stuff, you can kind of say, this is how I'm aiming, you know, to sample certain skeletons or to collect certain things from archives, and this is the plan. So it just depends sort of where you sit on the spectrum. So one way that we can make our sort of actual research process after you've maybe done a pre-registered report open is with open lab or notebooks. This is obviously less tidy than other publication methods, but the idea is to just digitize and share your research records, whether this is your diaries, your journals, your workbooks, your lab books, etc. I think historians of science are quite used to kind of seeing this, because a lot of notebooks of very famous past scientists are now actually open and fully scanned in, but people are kind of far more scared to do that now. I don't know if it's our imposter syndrome or people's perfectionism, but it's something that maybe we should be trying to do, because essentially the idea of having these notebooks and lab books is that if you were really ill and somebody had to carry on your work or, you know, do some of your stuff in the lab while you were unwell, they could just pick off exactly where you left off and can see what you were doing. So we should be making this more open and accessible. So the idea is to have free online access with reuse and redistribution type licenses. Being already talked about that, Emma's going to talk about that more. And ideally in a non-proprietary format, so not something that's requiring somebody to have some sort of software license to access. So if you can keep it in a plain text, markdown CSV, or, you know, just take JPEG photos of your lab book, which is something that I'm doing at the moment to try to make mine open, is just literally scanning them. And the idea is to document as you go, even your mistakes. So you can sort of link it to different things, maybe create yourself a template if you want to sort of do it all digitized from the beginning and try to standardize the way you lay it out. And to document any changes to the equipment that you use, any reagents that you use and software, so that if something maybe suddenly starts to make the data look a bit weird, you can work out what's going on and other people know what's happened. And of course, this can lead to accidental and serendipitous results. Penicillin is one of those absolute classic things of, oh, I just left them all in the sink. Oh, wait, antibiotics, amazing. And if they didn't have some of those things sort of documented about what they did when they went and then went away on holiday, we wouldn't know sort of what went on. But this is far less common than other kinds of registered reports or protocols. But I think this is something that's quite easy for us all to do. But obviously due to some safety concerns, so particularly if you're working with wet chemistry like I am, there's lots of acids and things, you can't physically have a computer or an iPad or your phone in the lab, and you certainly shouldn't be touching it with gloves. So you should have a physical book there that's sort of chemical safe, and then you can digitize it after the fact once you've sort of cleaned everything down. But something else that might be really useful is taking videos and stuff of what you do, or get somebody else to video you while you're doing your science. So it doesn't have to be a physical lab book, it can be a sort of video or photos of you actually doing the science. So this is kind of the most flexible format. Then of course there's lab protocols and other types of field methods. Like I said before, this should just be a step-by-step recipe guide like you'd find in a cookbook. Exactly what you do, listing everything that you use so that people could kind of find the same reagents or the same equipment and just copy what you did and hopefully get the same outcome. One of the ways that people are trying to make this open is using a website called Protocolsi. It's not the only way to do it. OSF and some of those other websites in our resources document also have an option for putting up your lab protocols and methods. But there's a really good Nature article that actually tells you how to make your own. You know, if you're starting from scratch or you're using something that somebody hasn't put up already, this is a really good step-by-step guide for how to document it and then upload it. So it's similar to the notebooks, and the keys are to document as you go, particularly if you're creating a brand new protocol. Take photos and videos of what you're doing, this can sometimes even help you write it up to go, ah, right, yes, no, I held it this way, or, you know, I shape it a certain way or something like that. Your institution might already have a template, maybe in another department, or you might have people that you collaborate with that have a template for some sort of protocol that you can use, or you can copy a template from somewhere else on protocols.io to help you, and just link to things that you use. So making it all open and showing people exactly where you get stuff from, and exactly the types of things you use can help with reproducibility. This is what protocols.io looks like. You can create a free account. It's got lots of different features on there, and this is going to be sort of one of the small steps I suggest if you're a lab-based person to sign up for, to put stuff on there. Here are some archaeological examples. We've got the Max Planck Institute are really, really great at putting theirs up on there. So we've got an example of their Zoom's pre-treatment protocol, so how they get the bone collagen out and how they treat it to do protein fingerprinting of archaeological samples. And then we've got an example of what they do for dental calculus. They've got a field sampling protocol as well as a lab-based protocol, so you can have a look at what they do, and they've got photos, as you can see there, of sort of literally kind of how they scrape stuff, and it's a step-by-step guide. And this is something that I'm hoping more isotope people will sort of put up there. But yet again, if you're not lab-based, don't feel like you have to do this. Just documenting what you do will be enough. But if you're lab-based, this is really great. Obviously, we do have some restrictions. So personally, I can't share the protocols that I use because that copyright's actually with my old PI, but she's hoping to put them up there. So it's a discussion to have with your lab group and your institution about how to sort of do this and put that up there. So study and research protocols are a bit different, where lab and field sort of method protocols are a step-by-step instruction manual for literally what you're kind of doing to dissolve the bone or look at stuff under a microscope. It's how the whole study was conducted overall. So that's including all of your data analysis that you're going to be doing with Guyon later, as well as anything that you do to prepare stuff before you get into the lab. So it's kind of everything encompassed as a whole, and it's to share not only your study design and analysis plans. So it's similar to a registered report, but it includes sort of lab protocols as kind of a subsection in there. So it's kind of more encompassing. Again, like I said before, clinical research people tend to do this, and then they have each sort of lab protocol to say what's going on there. But it's a good thing to sort of think about it like your whole grant proposal. And if you've written a big grant recently, you can kind of pre-register it or do a research protocol, and basically you've already got it all ready to go. Another way to do this is kind of already been touched upon, so I won't go into this in detail, but is metadata and data papers. So when you do your data paper, like we just saw before, document everything you did alongside it. So this is an example of one that I did where I had both meta-analysis, so it kind of tells you what I did to find the extra data that people had out there, but then I also say exactly what I did for the data that I did in the lab. So I literally kind of take everybody through, I use this method. If it was, you know, a big bone, I cut it down and I go through all of the acid steps and everything, but then I also say how and what I did to the published data to pull it into a bigger data set. Sometimes this comes as README files. Sometimes this is just a text file that you can download with a data set, but basically just document everything you did, and then people can work out what went on. So you can also obviously link this with stuff in traditional journal articles. So you can either link to a pre-registered or registered report, a methods paper, study or lab protocol, or you can just have it as your supplementary material. This is clearly the most common route, but I think this is changing. And I think we saw really, really good examples of workflow to do this, not at the end as supplementary material, but have supplementary material as just one part of documenting it throughout the whole process and having it open from the beginning. Because often, as we all know, there's strict word counts if you want to publish in a traditional journal. So it's really hard to make everything completely open within their word limits. But some of them are changing this, and I'll show you some really good examples of how journals are kind of letting you make your methods completely open in alternative routes. So there's the tier protocol, and the link's there, and you can all access the slides and click on that link if you want to. It's a really great sort of way if you're working, particularly in R&R Markdown, to kind of download a template to kind of keep all of your methods organized, how you should structure it. So if you're starting a new research project from scratch, you can have it all ready to go. It's mostly been set up to use in the classroom. So this is kind of really set up for smaller student projects, but I found it really useful in trying to structure some of my papers and things moving forward, because I kind of found it too late in my PhD to start it from the beginning, but it's something I've tried to work on since then. And it's particularly good because you've then got everything ready for when you do decide to publish. The people who did TIR also did the DRESS protocol, documenting research in the empirical social sciences. I don't think it needs to just be for empirical research. It's more tailored to sort of larger research projects. And yet again, it's a downloadable sort of template and guide to follow when you're setting up your methods and documenting everything that you've done. And they've made sure it's in line with most major journal policies, and it helps you sort of gather your supplementary material or to put it onto GitHub or OSF. It just has it in a nice neat package. And what they say you really have to have is a README file, which is kind of an instruction manual for how to use whatever you sort of upload in the repositories. The list of the results in the paper that you want people to reproduce, the things that are reproducible, because as we all know, some stuff might not be able to. Obviously, if you're kind of detailing your field methods, people won't be able to go back and dig the same site and get the exact same thing. That type of archaeology isn't reproducible, but you can say, I want you to reproduce my figures in this way, and this is the results that you can then run the code for. Any necessary software information, all of the data information that we've just heard about before in the previous talk, any command files people need to use. So this could also be linked to some lab stuff that you need if you need certain command files to kind of read what comes out of a mass spec, for instance. Any process data files. So again, I often have sort of raw data that comes out of the mass spec that then needs to be cleaned. So you can have raw and then the process data that then actually looks like what you see in a journal article, and basically step-by-step instructions for how to do what you did. This is very similar to what Cell journals have in there as well. So Cell, while most of us might not be publishing in Cell, they have a range of other journals. So there's quite a few that I've seen sort of ancient DNA and isotope work in particular, and they require this. So if you want to publish in a Cell journal, you have to have STAR methods. And STAR methods are structured, transparent, accessible reporting. And they dictate what sort of sections and headings you have to have. So this is yet again availability, so who to contact if they find any sort of issues with your work or to discuss it with you, where the material is, the data and the code, what type of experimental model and subject details you had. So yet again, this might not be needed for a lot of archaeological projects, and they will allow these things to be omitted if it's not applicable, but your method details, and they want it in excruciating detail. Then they want the details about your quantification and statistical analysis, any additional resources that you might need to link to and things. So this is the opportunity for you to link to an Open Science Framework account or your GitHub or Zenodo or whatever you might need, or your protocols.io, and a key resources table. So again, sort of highlighting anything that is essential to reproduce your results. And what they say quite specifically is your method should be detailed enough that readers don't need to go to other papers to understand what you did. So you can cite a methods paper. So for instance, I and most isotope people use a modified Longan method to get collagen out of bones. So it's not enough for the STAR method to just say, I used a modified Longan method, see this. You need to actually say, right, I cut the bone, I then cleaned it in this way, I put it in this acid for this long. They want you to actually put basically the protocol in there, and not just even link to a protocol. They want it in there to make it more fair and accessible for people to read it then and then when they're reading your paper, as well as then have links. So you can obviously cite methods papers to say, okay, well, this is being fully peer reviewed. I'm not using something that's completely off the wall here, but here is exactly what I personally did in the lab. So it shouldn't be a substitute for procedural details. And so I think I'm like, yeah, quite ahead of time here because I talk way too fast. But the small steps to start making your methods more open is to basically just start keeping all of your notes and trying to digitize them where you can. So scan in your notebooks. If you are doing things on an iPad out in the field, that's awesome. Make sure that it's backed up sort of as soon as you get back to the office. Register for an Open Science Framework account, because that's kind of the best way to keep all of this sort of collated and together. And go through and check the journals and platforms to see if they accept registered reports, or the kind of supplementary material that you would need to make your type of work more open. There's some really great tools that you can use to sort of check this. They're also really great to check some of the stuff that Emma's going to be talking about later about how open they are, about how much they're going to charge you or not charge you to do it. So you can use journal checker tools. And I'd really recommend having a look at the peer community in archaeology about how to kind of link all of this together and get this peer reviewed and out there. So those are some of those small steps. Yeah.
Thank you. I think you're about on time, don't worry.
I went way over time in practice, and yeah.
It's fine. We've got more time for questions, and I should stop there.